Hello, I'm Dr. Greg Ware. I'm the Chief Scientific Officer, co-founder of NeoGenesis. Uh, there's a lot of misconceptions about how molecules can penetrate through the skin when they're topically applied. You'll see a lot of stories surrounding exosomes from whether it's stem cells derived from human and animal tissue, plant stem cells, or platelets, the exosomes derived from these different cell types have various stories behind them. And particularly when you're talking about exosomes that have been isolated, whatever the source of the exosome is, as you isolate the exosomes, you're throwing out much of what Mother Nature has provided to us. Furthermore, when you do these harsh techniques to separate out the exosomes, you're taking away part of the penetration qualities of the exosome. So I'm going to tell you why the NeoGenesis Escort RM technology is the most advanced penetration system for skincare. The big hint is we don't disturb what Mother Nature gave us. Let me explain here. When we look at the secretome, that is all of the molecules that are released from a cell, and NeoGenesis takes all of the molecules released from multiple stem cell types and puts that into our product. If you look at the secretome, all of the molecules, there are three major components in the secretome. You have heard about the exosomes. And these exosomes are little lipid bilayer structures, like little balloons. And they're typically about 80 nanometers in diameter. And on the outside, if you obtain the exosomes correctly on the outside, they contain penetration enhancers. Now, there's also another vesicle type. These are called ectosomes, and these are derived naturally and differently from cells. And these contain a lot of the important molecules to maintain and heal the skin. And these ectosomes also contain penetration enhancers. These ectosomes are typically a little bit larger than the exosomes, and they contain a different variety of molecules than do the exosomes to help maintain and heal the tissue. So we incorporate all of that. We don't just isolate out the exosomes. Furthermore, and very importantly, there's a big soluble fraction in the secretome. That is all of those molecules that are outside of the exosomes and the ectosomes. These are molecules in the liquid portion of what's being secreted by the cells. This includes peptides, proteins, microproteins, lipids, and non-coding RNA signaling molecules. All of these molecules are contained both in exosomes as well as ectosomes, and then also in the soluble fraction. We don't throw any of that away. So we understand very clearly that if you use exosomes just as they are derived from the cell, released from the cell, the exosomes and the ectosomes contain these penetration enhancers. That means there's molecules on the outside of the exosome and ectosome that are going to allow it to make punctate holes in the matrix of the skin and push through the skin. They're opening little channels, they open and then close. And this way the exosomes and ectosomes can penetrate the skin. Well, interestingly, the soluble fraction, these molecules that are outside of the ectosome and the exosomes, they follow through. These molecules are going to flow through the channels made by the exosomes and ectosomes. This includes all the molecules I mentioned that are in the soluble fraction. That includes lipids. It can include some non-coding RNA. It can include proteins, peptides, and microproteins. All of these are kept in the Escort RM. We don't throw anything away. So a lot of companies who are isolating exosomes, 75% of the proteins are in the soluble fraction, and then another large number in the ectosomes. So the exosomes only contain some of the molecules. They're just a portion of the story. So this is why the S2M technology from NeoGenesis, I've been asked many times, how is it your products with the S2M technology work so well? And it's because of these basic components in the S2M that we keep things natural just as Mother Nature gave it to us. We got the exosomes, the ectosomes, pushing through the matrix, opening these punctate holes, which then close. And then the soluble fraction, the molecules outside of the vesicles, are flowing through these punctate channels. This is why the S2M works so well. You haven't thrown away any of the molecules, and because you haven't damaged the exosomes and ectosomes, all of the molecules can penetrate through the skin. So if you have any more questions about this, please reach out to me. I appreciate your time and interest.